Okay, thanks very much, and particularly for the invitation to come and talk, and the privilege of actually coming in honor of Sarah, because one of my things I remember most of all is her little visit to my laboratory on a fall day when there was a football game going on, and I think it was raining as well. She spent the whole afternoon trying to get spectra, and it didn't take her long, so she knew what she was doing, and it's a pretty incredible story. And Chuck McEwen was there too. And Chuck is one of my former students. Now, I don't often admit that, but it's okay. All right, so I'm going to change the venue a little bit, because what I want to talk about is what comes after the ionization. And what we'd like to do is see if I can convince you that it's possible to use Edmund sequencing and just read amino acid sequences of intact proteins on a chromatographic timescale. So the people who do this in my laboratory are Michelle English. So the two people back up here, Michelle English and Wei-On Wong, and this is Dina Bai, and of course Jeffrey Shabanowitz, who's been with me for a long, long time. So let me take you as a brief review of electron transfer dissociation that was discovered in our lab about 2004. And it was a response to Fred McLafferty and their discovery of being able to fragment proteins with electrons. And we wanted to be able to do it inside a linear ion trap mass spectrometer, and you can't do that with electrons because they can't store them. So we adapted this linear ion trap for electron transfer dissociation, where we do the chemistry with somebody who traps ions. So that's the front of the mass spectrometer and the front of our mascot, which is a hedgehog, and that's the back end of the hedgehog, and this is what we had to do to the back end of the linear ion trap, namely hook up a second ion source. And the people who did it were, of course, Josh Kuhn, who then was a postdoc student. He's now at the University of Wisconsin. And John Syka is an employee of Thermo and was also a graduate student in my lab after he was an employee at Thermo. So let's see if I can make this work. Okay, so how do you make electrons work inside a linear ion trap? You use chemical ionization in the gas phase and bombard gases with electrons, and that makes a thermoelectron. And then if you throw in an aromatic hydrocarbon, this is a particularly good one, it captures the thermoelectron, makes a radical anion. And now you can use that as a reagent inside the mass spectrometer. And if it walks up to a positively charged peptide, this is not an electron now, it's the radical anion of fluoranthene. It walks up and transfers the electron in the gas phase to the carbon-oxygen double bonds, eventually abstracts a hydrogen and produces a radical, and the radical then triggers fragmentation on either side of the amide bond, so you produce two pieces. One piece is called a C ion, and it contains the amine terminus plus one or more amino acids, and the other species is called a Z ion. It contains the C terminus of the protein or peptide plus one or more amino acids. And the important thing to note is that the only way you observe either or both of these two fragments is if there's something carrying a charge that allows the mass spectrometer to separate it. So that becomes important a little bit later in the lecture. And the other thing bad about this protocol, about the only thing that's bad about this process of chemically fragmenting proteins in the gas phase, is that proline is not a helpful amino acid. So if you transfer the electron into the amide bond that connects it to proline, the same chemistry takes place, but now you open up the ring system and the two fragments stay together. So capturing electrons into proline amide bonds uses up the electron, doesn't help you fragment the protein, and is therefore a negative effect. But that will be important also a little bit. Okay, so how did John Syka build this instrument, and how does it work? Basically, he divided up the linear ion trap into three segments, and then you just bring in your positively charged ions by electrospray or by Sarah's technique into this device, and then when you want to pick one to fragment, you just irradiate the others and kick them out of the trap, and so you store only one ion or one charged state of a protein, and then you move it over and store it in a well over here with voltages. And then you generate your negative ions from the back end of this device. Negative ions come in. There might be multiple things there, so you want to purify it by irradiating and kicking the ones out of the trap you don't want. You only want the one that will transfer an electron, not the one that will pull protons off, for example. And then after you purify your reagent, then you let them react together inside the linear ion trap, And now the negative charged particle transfers an electron to the positively charged one, fragments it. Now you're going to let the negative ions escape out this way, and then you scan the positively charged ions out to a detector. So you have fragmented your protein or peptide by doing a chemical reaction in the gas phase. And the whole process doesn't take very long. It goes just as fast as the collisional processes that everybody else uses at the moment. The neat thing is that you produce a picket fence kind of spectrum. This is a it's got a doubly phosphorylated peptide, but you note that there's also 2, 3, 4, a total of 5 possible sites for these two phosphates, but it's totally unambiguous because this process does not affect post-translational modifications. It just breaks the amide bonds and disulfide bonds. Okay, so that's old stuff. I should point now we're going to go on to look at proteins, but in the meantime, this was the second paper, I guess the third paper we published, second paper we published on this. And we showed that you could use this device and use this chemistry and couple it with another one of Scott McLucky's ion-ion reactions, which we'd like to use all of his reactions, but so far we've only used 2. Anyway, the point is, if you want to clean up a spectrum that is very complex and simplify it, spread it out so none of the ions overlap with charge in the same space, then it's possible to bring in a second reagent. In this case, it's the anion of benzoic acid. And now what you do is do proton transfer reaction. So this is ubiquitin on a linear ion trap, and we can't resolve anything past +2 in a linear ion trap. So anytime we have +3, 4, 5, 6, 7, 8, 9, 10, 11, and 12, those fragments are all grouped together here, and I can't resolve them. So what we learned, however, was that if you bring in this anion of benzoic acid, it'll start pulling protons off these species, thereby reducing their charge, therefore spreading the ions out. You'll lose a lot of them because they'll exceed the mass range of the instrument, but those that are left behind are simple to interpret and highly desirable. And so now we're going to change the time. We're going to do 15 milliseconds of this electron transfer to make that fragmentation mess, and the fragments are anywhere from +12s down to +1s, and they're all mixed together. Now if you bring in the second reagent, which is the anion of benzoic acid, and we let it react for 50 milliseconds, you can see that already and this is just blowing up those little peaks. But the point is, I pull protons off these peaks, the charge state goes down, and I spread out the ions over the entire mass range, and the ones that had lots of charges on them get thrown out beyond the mass range. And you can control this reaction because the rate of the ion-ion reaction in the gas phase goes as the square of the charge. So, for example, if you have a +10 ion, it's going to react 100 times faster than a +1 ion. So if I have a long reaction time, I'll destroy all the charges and go down to +1 and 2. If I do short reaction times, I'll save the multiply charged states, and I can let them grow into the spectrum and easily interpret it. So we're still on a low-resolution instrument. I just wanted to show you what happens, and then we're going to go on to a high-resolution instrument with some new technology. Okay, so if I let the reaction go a little bit longer, 15 milliseconds of fragmentation to make that, but then use 100 milliseconds of time with the anion of benzoic acid, it's still getting simpler and simpler. And I'll go one more step, and this is what happens if you let the reaction go for 150 milliseconds. The end result is all these charge states have been converted to +1s, and the only thing left are a series of ions, but they're key ions because the C ions are the ones that contain the amine terminus plus one or more amino acids, and you can see there's C2, 3, 4, 5, 6, 7, you go all the way out to 17. So you've read off the first 17 amino acids of the protein, whatever it is, and the Z ions are the ones that contain the carboxy terminus of the protein, and it starts at Z3, and from Z3 you can read 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way out to 17. So in an experiment that takes 100 milliseconds, you've just read the amino acid sequence from the N terminus and the amino acid sequence from the carboxy terminus in 17 amino acids. You can't go any further because the mass range of this instrument is 2,000. All right, so you know the molecular weight that you started with from the electrospray spectrum. You know the amino terminal sequence, and you know the C-terminal sequence. You can identify any protein in the database. And if the molecular weight that you observe of the protein in the database is the measured molecular weight is higher than that one, then obviously you would conclude that you've identified the right protein, but it must be post-translationally modified. And now you go in and look to see what that modification state is. If you happen to find that the molecular weight you've measured is less, then you've probably got a splice variant. The point is, in a very short period of time, you can identify proteins very quickly from this information. Okay, so then we took it on one more step, and a third paper. And this one, I don't think anybody's read, but the point is it was pretty neat, and it was in my birthday issue of International Journal of Mass Spectrometry. But we got a sample from Sandy Markey's lab, and basically consisted of ribosomal proteins, 21 from one subunit, 33 from the other, and we just asked the question, is it possible to identify all those proteins chromatographically on a usual electrospray ionization nanoscale protocol? And the answer is yes. And so this is what the spectrum of a bunch of those proteins looks like, doing a C4 column. And basically you could take each one of those peaks and get the first and last 17 amino acids and then identify all those proteins on a chromatographic timescale. Okay, so in summary, and we call this now ion-ion proton transfer to indicate that it really is an ion-ion reaction that we're playing with, as is ETD. It's electron transfer, and it's an ion-ion reaction. In any case, you could identify all those proteins. We found a number of them were post-translationally modified, and you could figure out what those modifications were. So, so at the moment, then, I would say you can identify lots of proteins on a chromatographic time scale with that information. Now, the next challenge is, of course, not only just to identify the protein, but be able to characterize everything that's going on that protein. And so it was obvious that we needed to hook the linear ion trap up to one of the other analyzers, like the Orbitrap or the Fourier transform instrument. I didn't have an Orbitrap. I had the Fourier transform instrument in my lab, and so we decided that the best choice would be to modify the instrument so that you would have the electron transfer dissociation being introduced and negative ions being introduced on the same side as you do the electrospray. Unfortunately, my student Josh Kuhn decided that he would go build the Orbitrap instrument and introduce everything from the back end, which I think was a big mistake. But it became commercial. So the point is, what he's doing is generating his fluoranthene radical anions back here. He sends them all the way through this device to the linear ion trap, and then he fragments the peptides or proteins, and then he sends them to the C-trap and sends the ions into the Orbitrap. And I'll show you in a minute why I think that was a bad idea. So this is front-end electron transfer dissociation, where we have modified the ion sources on the Orbitrap. They don't look too differently. And the bottom line is that if I go back to this device, this is actually enclosed in a box that's about the size of this table. And it requires pumping and optics and all kinds of other stuff. And then you go over to the front-end electron transfer dissociation. All it is a little device that sits on the inside of the ion source, and it's about the size of an eraser on a pencil. So that's a huge plus. Then we switched over to azulene and fluoranthene. They're about equal. This one goes 3 times faster. And it vaporizes at atmospheric pressure. So it's probably the reagent of choice, although fluoranthene is still usable. All right, so now we go over to the Velis instrument, which has the Shrig here, and that turns out to be quite nice because just past the Shrig is the right pressure for a discharge source for chemical ionization. So as you'll see in a minute, we have built the source for anions directly on the other side of the Shrig, and now it's possible to do ETD from the front. So this part of the mass spectrometer can disappear altogether over there. So this is how it works. So there's the little Shrig, and it's just part of basically a lens structure, but it has a gaseous discharge in it, and it'll be pulsed on and off to create negative ions whenever we want them. So electrospray ionization comes in from the front, or Sarah's technology, and this would work with her technology as well. And so the electrospray ionization sends the ions over to the linear trap, and whenever we want negative ions, we just turn on a pulse here, and it creates our negative ion reagents to go in and do the chemistry that we want to do. So electrospray ionization, transferring ions to this linear ion trap here, storing them, selecting which ion we want to fragment, parking it over here in the end of the chamber, And now you go back and generate negative ions, bring those into that linear ion trap, pick the ones you want to do the chemistry, and then let them react with the ETD positive ions to produce fragment ions, and then you scan those down to the Orbitrap to be analyzed under high-resolution conditions. If you want to do proton transfer on this instrument, then you just switch over. Both reagents are coming in at exactly the same time. You're just mass analyzing and picking the one you want to do the chemistry. And so you send your proton transfer reagent down here and pull the protons off the ETD reagents and then send them on down to the Orbitrap. Okay, so the other huge advantage of this instrument is the following, that I can do, if I do ETD or proton transfer here on protein ions, and I bring them down here and fragment them and change the charge state and spread them out and do whatever I want to do with them, I then transfer them down to the C-trap, but then rather than analyzing them, I can simply pick another set of ions over here, put them in here, do the same chemistry on them, park them down in the C-trap. In other words, you can do multiple fills of the C-trap before you ever record the high-resolution mass spectrum. Now, many people doing top-down work at the moment in the field actually average transients, or they average the results they get from a particular spectrum. And to get the right sensitivity, they're sometimes averaging 100, 200, 300 scans, or in a recent paper, even 20 HPLC runs, just to get enough ion current to do the mass analysis of an intact protein. But when you average transients or you average spectra, the signal-to-noise you get goes as the square of the number of scans that you took. So you have to take 100 scans to improve the signal-to-noise by a factor of 10. In this case, we don't average anything. If we want 10 times more ions, we just do this experiment 10 times and park them all down here one after the other and then finally analyze them. So right off the bat, you gain a factor of more than 50 or 100 in sensitivity. Okay, so now I'm going to illustrate what we're trying to do on a model protein, which you've already seen, which is apomyoglobin. But we're obviously after 50,000 molecular weight proteins, including therapeutic antibodies. So the protocol here is to be able to analyze a protein on a chromatographic timescale, that is peak widths of 30 seconds or a minute, and get down into the femtomole level and be able to get you not just the identification, but to get complete sequence coverage. And if I have my way, I want to read the sequence from the N-terminus all the way to the end, and I want to read the sequence from the C-terminus all the way to the end. And in fact, do it so you can look at it on a piece of paper and do it yourself if you want to. That's what I'm spending hours doing at the moment. Okay, so it's going to have front-end electron transfer dissociation. We can alter the abundances. We don't have to average anything. We're just going to have multiple fills on the C-trap, and we can select the ions that we do the multiple fills on. So we don't have to put all the ions down there, just the ones we want to interrogate. And then we develop some chemical derivatization techniques to enhance both the fragmentation that we want to see, localize it, and at the same time enhance the charge on the protein that we're analyzing. And then we'll use the ion-ion proton transfer reaction that I've already showed you to simplify the spectrum, prevent overlaps of multiply charged peaks so we can spread them out and have no difficulty interpreting them. And then finally, the last step, of course, would be to deconvolute all these spectra so they can print them out on a piece of paper that runs from here to where Chuck is sitting, and it just has one mass all the way up to 50,000. All right, so here's apomyoglobin. It's a standard protein. It has 153 amino acids, 17,000 molecular weight. 22 of them are basic, and 20 of them are basic, and 22 of them are acidic. And they're pretty evenly distributed. So if you want to play with a model, this is a pretty good one. So here's the experiment. We're going to do nanoflow HPLC. That's 60 nanoliters per minute, and that's based on technology we developed many years ago. And we can alter the gradient so that we can spread our protein peaks out to 2 minutes if we want to, for practice or for changing variables. But the point is, we would normally work with something that's about 30 seconds wide. And at the moment, we're operating at, on this protein, you could go down to 10 femtomoles. Okay, and then C-trap fills. It takes 50 milliseconds to fill up that C-trap with the number of ions, whichever ones we want. You can increase the signal by 10 to 20 by just filling it up 10 or 20 times. The only cost is time, and that can be done in about 5 seconds. Transients also take time. If you operate at 100,000 resolution, then it takes you about a second to do that. On the new Orbitrap, Makarov just put out, this time it gets reduced by a factor of 3. Okay, now the time to require 5 millisecond ETD spectrum and do PT proton transfer and everything is probably on the order of 5 seconds. At the moment, it'll get faster, particularly if we have the new Orbitrap. Then the whole point is, on a chromatographic time scale, I've got time to do about 10 experiments. Any 10 experiments I want to get at the sequence information. So here's a typical protocol. We're going to first acquire the molecular weight of the protein, just electrospray ionization, high-resolution mass spectrum to define the molecular weight. And then the next thing we do is record an MSMS spectrum, 10 to 20 trap fills if we want, 5 milliseconds of ETD with fluoranthene, and that'll give us, and then usually 100 to 150 milliseconds of proton transfer. The reagent of choice at the moment is SF6. That just requires like parts per million of SF6 to do this incredibly fast reaction. But if you let it react for that period of time, the spectrum is just going to contain +1 and +2 ions. And that should let you, because we have an expanded mass range now of 4,000, that should let you read off 60 amino acids or so at each end of the protein in a pretty fast time scale. So here, let's go see what it looks like. So that's an electrospray ionization spectrum of apomyoglobin, and we're going to play with this +25 ion here. We can pick any one or multiple ions to play with. I'm just going to illustrate it with this one at 679. And that's what you'd see if you did 5 milliseconds of ETD. You totally destroy the whole protein, and you produce a huge fraction of positively charged ions that have charges up to +10s, 15s, and 16s. But they're all in the same mass range, and they all overlap. And even with 100,000 resolution, you can't go in and pick them all out. So now we'll do 5 milliseconds of ETD to produce that same spectrum, but then we'll let the SF6 anion come into those ions and pull protons off them, so that after 160 milliseconds of ion-ion chemistry, the only thing you're going to see are +1 and +2 ions. And you note that it begins with the C3 ion. Remember, that's from the amine terminus, and we're missing the fragment that would tell us what the first 2 amino acids are. It starts at C3, and the only thing, that's because the RF used to transport the ions down to the C-trap cuts off that low mass portion of the spectrum. So we're going to want to have a cure for that, and I'll show you in a minute. And then if you go to look at the Z ions basically start at Z7. So we're missing the last 6 amino acids. We find the fragment that contains 7. And that's because the C-terminus of a protein is usually not basic, and there's no way to carry a charge. So we've got to fix that too. But for the moment, this spectrum gets us basically C's and Z ions, +1 ions, and +2 ions. Okay. I'll just point out that. So the reason here you don't see the Z's 1, 2, 3, 4, 5, and 6 is because these residues are not basic, and we don't see the first 2 amino acids up here because their mass adds up to something that can't be transported down to the C-trap under the conditions we're running the spectrum. Okay. So now, keep going. This is just the rest of the mass range after 160 milliseconds, and you can see the +2s and 3s growing, and now we're out to 27, I think. And if you carry up to 2,000, you're getting up to, and it's hard for me to see, but in the 30s somewhere, I think. And if you go up to 2,500 and 4,000, now you're reading up into the 50s with a spectrum that's easy to interpret, and you can do it manually by just looking at the spectrum. And since we're doing it at high resolution, of course, these isotope peaks are all 1 mass unit apart, or 1/10 of a mass unit apart, depending on charge. Okay, so where are we now? That's the end of the scale. Okay. So now let's solve this. First, let's solve that problem about not being able to see the last amino acids and the first amino acids. So we can really do Edmund. We got to find those. So that we're going to repeat that experiment, but we're going to derivatize the proteins so we can identify those initial residues at the N and C terminus. So the chemistry we do is this. So we're going to derivatize the carboxy terminus of the protein and every other amino acid that contains a carboxylic acid side chain, which is aspartic acid and glutamic acid. And the point is, if I convert that carboxylic acid that's at the very end of the protein into an amide, but the amide contains a basic residue, now all the amino acid fragments from the C-terminus back are going to be positively charged. So that's what we're going to do. And note that this is glycine with a hydrogen as its side chain. Its mass is 59. If I could put a proton on it, it would be 60. But I can't put a proton on it because there's nothing basic. Now I can over here, and since what I've added weighs 143, the mass of glycine is going to jump to 203 because I've made the mass heavier and I've given it a charge so I could detect it in the mass spectrometer. So the chemistry is this. You do carbodiamide chemistry. You take a carboxylic acid, about pH 5, and you protonate one of these nitrogens. That makes the anion come in and attack the carbon. Then after you've form this structure, you protonate the other nitrogen, and now the amine terminus of this derivative attacks the carbon-oxygen double bond and forms an amide and splits out urea. Now, the urea has got an amine on it, so it gets protonated and flows right through your HBlC column, so you never see that. But now you have derivatized the protein, so that the C-terminus is now an amide, and the amide is basic because of the histidine ring system. And you should have derivatized a lot of the other carboxylic acids. Now, most of the time, organic chemistry on intact proteins doesn't go too well because the protein is folded up in solution. But the thing that's usually true is that the carboxy and C-termini, the N-terminus and the C-terminus of a protein hangs out and flops around. So while we don't expect 100% yield, we should be able to quantitatively derivatize what we want to see. So here's back to the electrospray spectrum of apomyoglobin. And now that's what happens after you chemically derivatize it. It's a mess. And that's because we don't have 100% yield. But the point is, you can take any window of ions here, isolate them, send them in the mass spectrometer, do ETD on them, and this is what pops out. Absolutely fantastic. In other words, this glycine is now basic, and its mass has been shifted by 143. So it shows up in the spectrum right where we expect at 203. And then every other Z ion shows up because here's another one that's been converted to a basic residue. And then doubly charged and triply charged ions. All these Ds and Es have been converted by the chemistry. And now you read in one spectrum the first 39 amino acids at the C-terminus of a protein. And it does go further. So easy chemistry allows you to unambiguously identify the C-terminal residues, and that's what we need to do to complete the sequence. Now, the other problem was the first two amino acids at the N-terminus of the protein, and so we use this chemistry to get those, if we need to. So here's the amine terminus, and if it happens to be lysine, then the side chain is a hydrogen, and here's the protein going down this way. If you let that amino group attack that carbon-oxygen double bond and displace the hydroxysuccinimide sulfate, then you produce this tag species, and if you let it then react with aminoethyl maleamide, then the sulfur adds across the double bond, and now you've tagged the amino terminus of the protein such that you've increased the mass of whatever residue is there by 228. And if it's lysine, if it's glycine, it would be 303. And sure enough, that's what the thing looks like as electrospray without chemistry. That's what it looks like after you do the chemistry. Pick a window out here, put it in the mass spectrometer, fragment it, and now, sure enough, you see 1, 2, 3, 4, 5, 6, 7, and you read off the amino terminal sequence of apomyoglobin or any other protein. Okay, so now we've got the amine terminus and the C-terminus, and we're reading in so far about 30 or 40 residues. Let's repeat that ETD spectrum on the derivatized or underivatized material, but change the proton transfer time so we make it shorter. And if we make the proton transfer time shorter, we'll leave +3s and +4 behind in the spectrum, and we'll just watch those ions grow into the spectrum we recorded the first time around. And so here it is. Everything in black is what we already knew from the long proton transfer time, and those ions are still there. But now by shortening the time down to 80, all of a sudden we start seeing doubly and triply and ions with +3s and +4s grow in. And if we go all the way to the end of the mass range, I think you can see that this gets you up into the mid-60s. Okay, we'll change the time a little bit one more time. So now we're going to change it down to 5 to 15 milliseconds and the proton transfer time, and now you'll see +6 and +10 ions, anything grow in there. Remember, what we already know is in black, and the only new ones that grow into the spectrum are in red. And there you go, it gets more and more complex, but the bottom line is, at the end, you're up into the 80s, 85 or so. All right, so on that set of experiments, this is what you get out of the whole thing. So this is apomyoglobin, starting off at residue 1 at 150, and then it's 153 mass units long, residues long. So I'm going to read the amino acid sequence over several slides from top to bottom, and then we'll come back and look what happens on the other side, which are the Z ions. And these are just the reaction times that you use to do the proton transfer and simplify the spectra. So what we did initially was long proton transfer, and we see singly charged ions. We couldn't find the amine terminus because these two guys weren't there, so we did chemistry, and we filled that in, no problem. And now we keep reading along and get further into the sequence. The singly and doubly charged ions are reading off most of it, but the shorter reaction times in pink and blue begin to grow in. And by the way, whenever you hit proline, then you'll see a blank. If I can find one right there. So that's a blank because it's right next to proline. So we count that as being seen, since we know if proline's supposed to be there, then the next residue has to be histidine. All right, so +4s, 5s, 6s, and 7s growing in, and they grow in until you get up to about 85 or 86 residues in. Now if you go down the bottom and look for the other end of the protein, here's the amino acid at the C-terminus, and remember we didn't see the first 6, but we did chemistry, and then we saw the first 6 amino acids, so we know what they are. And now go backwards up, here's +1s, +2s growing in, complete sequence coverage, and then with the shorter reaction times of 80 and 40 milliseconds, the others grow in up to +7s, and again, when you work your way all the way back, we're seeing ions up and around 83, 84 amino acids into the chain. And now they begin to overlap with what we see on the C-terminus as well. Okay, so the bottom line is, if you take a look at this protein, this is what you get without chemistry. You get about 80%, 88% of the sequence covered. If we do chemistry on the thing, we got everything here and here. We're only missing a few residues in here, and we get 93%. We now know how to get these others, and that trick is done by multiple fills of ion packets that contain these missing residues, and they pop right up out of the structure. The reason you don't see them is because they're overlapping with higher charge states, and we can now solve that problem with multiple fills and doing chemistry inside the ion trap. I'll skip over the deconvolution, and I'll just give you some references here. So ubiquitin has shown up several times. There's 100% sequence analysis of ubiquitin. That is, we say we cover every amino acid going from the N-terminus and every amino acid coming from the C-terminus. Apomyoglobin, we've now got 100% of that as well. Carbonic anhydrase is about 29,000, and we get more than 90% of that at the moment. And we've also begun sequencing antibodies, unknown structures, and we get more than 90% of things like the light chain. So the earlier part of the talk said we can identify proteins using N-terminal and C-terminal sequencing plus molecular weight on a chromatographic time scale. Now, I think we're getting close to getting near complete sequence analysis of all amino acids and their post-translational modifications on a chromatographic timescale, and that's what's ahead of us in the next few months. So with that, I'll thank you for your attention. Say one more time how happy I am that Sarah got her award, and many more to come, and thanks a lot. Bye-bye.
We have time for one question.
Yeah. With 100,000 resolution, it seems like you should be able to resolve most of the fragments for myoglobin, and so I'm wondering how much additional information did you get by choosing the 3 or 4 different proton transfer reaction times versus not doing anything
If you think you can do that, you're a better man than I am, because I've looked at those spectra and it doesn't work. And the computer can't make it work either. This is much simpler, much simpler. And particularly when you start looking for post-translational modifications.
Are you going to discover new sequences that weren't expected with this method?
Sure.
Once you have
This is de novo. This is de novo sequencing. We'll figure out any structure of any antibody.
Do you see that when you look at something like ubiquitin? Is there a small, very small fraction of ions that are not quite what you would expect, but have the same reversal mass?
That's not, well, we have not investigated ubiquitin. We're using it to develop the technology, but not sequence it. But yes, you could. Now again, my dynamic range, so if there's a point, well, I don't know, but I mean, if you look at the intact protein, I don't have the same dynamic range that you do, and I won't have the same dynamic range I'll have as with peptides. So I've got to, I'm sacrificing dynamic range to sequence deep into the protein to find post-translational modifications. So that's the goal.
Thank you.
Okay. Can I pull out?